Hey friends, Dr. G here. Let me grab the door. Yeah, you'd think that before I hit the start button, I'd shut the door. Still, online, whatever. So, first exam has been graded, and we'll be posting that to the Moodle Gradebook Biz. This is the first exam. We're going to be, looking for a few other exams. 3 more. We're getting to them. There's an exam that we probably are going to get to next week, not this week. We have to go through microbial diseases a bit, in chapters 9 and 10. Anyway, I thought what I'd do today is go through the exam a bit, and I haven't figured out exactly how you're going to be allowed to pick them up, but I think a general thing would be Monday, Wednesday, Friday from 10 to 11, guaranteed to be here. Come by, you can pick your exam up. If you need to have a different time, holler. I can come up with something. If you have class, I don't know if everybody has A&P at 10 to 11 or something. Lectures 11 to 12. I often am doing a lecture at that point, but if that works, we can make that work too. I'll email you some stuff. So let's just go ahead and say for now, Monday, Wednesday, Friday, 10 to noon, come by and I can get you the exam. Regardless, what we're going to do is just briefly go over the exam, what I was looking for, and a couple of, there's one question that I think we're going to throw out, so we'll see how that goes. Anyway, we're starting out, you can pull your exam up and go over it. The set of fill in the blanks, they were 4 points each, not 4 points per question, 4 points each. And, okay, so the first blank, things decay because of life that wasn't originally visible. The idea would be that it is spontaneous generation, which we of course know is not true. Pasteur disproved that. Then when we start talking about the development of making polymers from monomers, we generate polymers by linking together monomers in what's known as a dehydration synthesis. You generate a molecule of water, which is going to be a multiple choice question here later. We break these polymers apart by hydrolyzing them, by adding water across the bond, so they're hydrolysis reactions. Because they don't have a lot of internal structures, prokaryotes or bacteria, cells can divide rapidly and exploit the multiple different environments. The many internal membranes of the eukaryotic cells force them to divide more slowly. Question 4. I think the best answer here is the ribosome is the only organelle that is in common. Bacteria would have a nucleus, or prokaryotes would have a nucleus, if they wrap their DNA in membranes. Question 5. The most important feature for determining what gets in and out of a cell is the cell membrane, that is essentially the gatekeepers of the system. Cell walls are keeping bacteria from popping due to osmotic pressure. Question 6. An organism that cannot detoxify the poisonous byproducts of oxygen would necessarily be an obligate anaerobe. That means that they cannot use oxygen, and oxygen is poisonous, so they are obligate anaerobic organisms. On the other hand, cells that prefer to use oxygen but can grow, often slowly in the absence of oxygen, this would be a facultative anaerobe. So while they are happiest with air, they can't operate in the absence of air, so that would make them a facultative anaerobe. Then we get on to multiple choice questions. So question 1, swan neck flasks allowed air but not bugs into the flask, that's E. Koch's postulates would work best for bacteria, but also for viruses. These other things are not actually things that you can actually extract from the organism to demonstrate and then pass it on to another organism or culture it in a way. Let's see, the eukaryotic cells, fungi are eukaryotes. Everybody else is either not a cell like a prion or a virus, or they are prokaryotes. Archaeal cells, the archaebacteria, these are the extremophiles, and so they're always going to be living in the more extreme environments like the hot springs. Everything else is a reasonably conducive environment, your colon, potato salad, the ocean. Let's see. Question 5 is the one I'm throwing out. So you're going to have a grade on your test, but I gave her by 2 points. Which of the that's in the grade book, it'll be there. Which of the following structures most resemble the complexity of the human body? We're looking at some sort of multicellular type of organism system. And the idea of a biofilm is where you have multiple cells interacting together as a community. That would be the best answer. That's D. A lot of people were saying things like cell wall or glycocalyx. And as I read this, I'm like, no, man, that's D. But I'm willing to let that one slide. Sure, bump question. All right, ions of an element, they've got an unequal number of protons and electrons because that's what makes them either positively charged or negatively charged. That's E. The number of neutrons vary. That's what makes an isotope, is the number of neutrons doesn't equal the number of protons. The number of protons determines an element's identity. Most stable bonds, that's going to be covalent bonds. Let's see. Things that, this is question 9. What can't speed up a chemical reaction? The first 4 items, heat, enzymes, catalysts, increased reactant concentrations, these can speed up a chemical reaction. Cooling it down, no way. That's going to speed up a chemical reaction. Let's see. So question 10. Molecules of water are produced when any biopolymer is made, and that's because of the dehydration reactions. 11. Proteins include all the following elements in their structure except for phosphorus would be the best answer. Now, you can have phosphorylation, but that's not really the same idea as the elements that are in the structure in terms of the other answers, because all amino acids are made with carbon, nitrogen, hydrogen, and they all have a peptide bond. 12. So in comparison to prokaryotes, eukaryotic cells, primarily the best answer there would be peptidoglycan, because all eukaryotic cells have a cell membrane, cytoplasm, chromosomes, and ribosomes. Peptidoglycan, as the sort of sugar candy coating, is really something that is limited to bacteria only. Okay, let's see. Question 13, found only in prokaryotes and present in all prokaryotes. The best answer there would be the nucleoid. Not all of the bacteria are going to have peptidoglycan. And then the other 2 things like flagella and cilia, ribosomes and cytoplasm are both shared in both eukaryotes and prokaryotes. The significance of the archaea in medicine is they're not very significant. That's the significance. They cause no disease. Bacteria of the genus Mycoplasma, they actually are very weird microorganisms. They don't have a cell wall, so that makes them gram-negative. And it also means they don't have a peptidoglycan, which would be part of the cell wall material. Question 16, the dichotomous key. That's what we use to identify a species through this yes-no testing. Question 17, extreme halophiles. Halophiles are guys that really love salt, and these are able to live in very high salt environments, or a hypertonic environment. B is the answer. Eukaryotic cells, of course, this is question 18, have a linear chromosomes. The lag phase of growth, this is where we had the beginning of the growth phase, the lag, the log, and then the plateau. At the lag phase, cells are adjusting to the new medium without dividing. Where would you least expect to find the archaeobacteria growing? Well, you're going to look for the least, or I guess the most hospitable environment. That would be where you would least expect to find them. So the top of rich Illinois farm soil, that's a great place for bacteria to grow. Archaea aren't going to be surviving there. Let's see. So if you get electrons from organic material, carbon from carbon dioxide, and energy from the conversion of hydrogen sulfide to sulfate, you're a pretty weird organism. You're not anything like me. The best answer there would be you be a chemoorganoautotroph. Okay. So question C, basophiles is neutrophiles and acidophiles. This is referring to organisms that can grow in a range of different pHs. Answer is C. Most human pathogens are able to grow in an environment that is probably going to have a fair amount of oxygen because we're fairly oxygenated in most places, but there then are going to have some areas that don't have a whole lot of oxygen, the colon and various sorts of places in your sinuses and things. The best answer there, though, would be they're going to be facultative anaerobes. They prefer to grow in an environment with oxygen, and they probably need to be able to grow in an environment with oxygen, but they are able to grow in an environment without oxygen. All right, so then the microassays, just truth in advertising, because of the way the grade distribution was perfect, I stopped here. And so most people got these correct. If you didn't, we didn't nick you down too hard for these. But the importance of the microscope, it allows for the visualization of very small structures. It develops greater resolution in being able to visualize cells. Monomers and polymers, they are related. A polymer is made up of monomers. Monomers are the repeating subunits that make up polymers. The long fatty acid tails, these are providing the nonpolar characteristics of a amphipathic molecule. So you have the phosphate head in a phospholipid, and then the long nonpolar tail, and the long fatty acid tails then end up being critical for the formation of micelles and also for the formation of the phospholipid bilayer. The lack of many membrane-bound organelles found specifically in prokaryotes. So prokaryotes don't have membrane-bound organelles, which makes their cytoplasm significantly less complex. There's less compartments, so it makes their ability to carry out chemical reactions and biochemistry a bit more simplified, but that's a critical point between the primitive prokaryotic cells and the more advanced eukaryotic cells. The stationary phase of the growth curve, this is the top phase of the growth curve. The lag phase is the beginning. The stationary phase is where the number of bacteria is remaining somewhat constant in that the growth is being offset by the death. Chemolithal autotrophy, this is a growth habit in which they're using chemicals and minerals as their electron source and energy source, and then autotrophy, they're able to actually bring carbon in by capturing carbon dioxide. These are really weird microorganisms. Hey, that's the exam. We're going to get back into lecturing and see where we go. So what I'm going to do is type up a little bit of an assignment for you to find an article that is of relevance to you. Honestly, I haven't decided whether I'm going to give you the article or have you go out and look for one. We'll see how that goes in the next few days. And this will be some of the points that we can kind of add on, so it's not just the exams that are making up your grade. Hey, that's all I know for now. Yeah, that's it. So in terms of picking up your exam, come by Monday, Wednesday, Friday, 10 to noon guaranteed. Hit me up if you need a better time or a different time, and I'll accommodate you as well. Honestly, 10 to noon on Tuesday, Thursday is pretty much of a standard time to find me as well. I'm trying to be up here as much as I can. All right, so that's what I have to say now about that. And that was exam 1. Let's get ready for exam 2. We'll be finishing the material up for that this week and get ready to jump into it next week. And we may change the exam formatting up. This was a little bit of a pain to grade, but I'll let you know how that goes. All right, guys, thanks for your patience and your participation, and we'll see you around. Bye.